Good afternoon. Welcome to our Thursday afternoon seminars. This is sort of a return to in-person seminars for us. We haven't had one in a little while. So thank you all for coming out. We are extremely lucky to have Dr. Justine Becker with us here today. She has come to us all the way from Montana State University in Bozeman, where she has the terribly difficult task of taking on a new position as assistant professor there. A terrible place to have to live. We're all very sorry for her. Justine is, I would say, relatively rare among scientists in being undaunted and quite skilled at both field ecology and doing really serious quantitative modeling. So I invited her here because I thought her expertise, particularly in movement ecology, would be of interest to a large number of people here, given the work we're doing on the Florida Wildlife Corridor and animal movement, Joe's work with the Corridor Observatory and camera trapping, and of course, all the movement work we do with gopher tortoises and several bird species. So I would encourage all of you who are interested in those sorts of topics to engage with her after the seminar. Justine has a deep background in wildlife management and wildlife biology and ecology. She began her career as an undergraduate at University of Auckland in New Zealand, where she hails from. And I understand did some individual-based modeling of fragmented landscapes there. And I need to ask her, perhaps at dinner tonight, how it is that she has a double bachelor's in biology and history, and then apparently 1 year later, some sort of biology honors bachelor's, which must be a New Zealand thing. That's right. In any case, she then moved from there to a PhD program where we crossed paths. And she was a great collaborator and led several studies on African antelope movement, which I'm sure you'll hear about today. That included some really creative coupling of camera trap studies of antelope behavior with both GPS data tracking of movement, and then some really computationally advanced basic ecological studies of niche partitioning among closely related species of antelope. As a postdoc at University of Wyoming for 2 years, she started into some pronghorn migration work, which I gather we'll see some of today as well. And it sounds like in her new position at Montana State, she'll be diving in to a lot of conservation relevant work with the state agencies as well as continuing her basic ecological work. So thank you, Justine, for making the trip all the way out here. It's been a great visit so far, and I'll turn it over to you.
Well, thanks so much, Josh, for inviting me and for that introduction, to you all for joining me today. I'm really excited to talk to you about a bit of my research. So I wanted to start today actually with just a quick short story that starts with a weather event that you may be more familiar with since you live in Florida compared to me in Montana. But this actually happened all the way over in Southern Africa in Mozambique. So in March of 2019, Tropical Cyclone Idai made landfall over the city of Beira in central Mozambique, and it remains the worst storm on record in this area of the Southern Hemisphere. It was absolutely catastrophically devastating to this region for all of the human communities living in this area. And from an ecological perspective, it also had a really major impact on some of the ecosystems. So the cyclone actually passed almost directly over Gorongosa National Park, which is highlighted in that little black blob. Hopefully you can see that there, which is in central Mozambique and is home to a really interesting and diverse community of large mammals. And I'll get into a bit more information about Gorongosa later in the talk. But at the time that the cyclone hit, we were actually conducting a lot of different research on these mammal communities. And we actually had GPS collars on several different species from different populations in the park. And so we had a really unique opportunity to monitor in real time how these animals were responding to this catastrophic event, which we felt was going to be particularly interesting because this kind of cyclone is going to increase in intensity and frequency, as predicted, with climate change over the next several decades. And some pretty dramatic flooding took place in Gorongosa. So this is one of the structures on the floodplain in the park, and this is prior to any flooding occurring. And this is what this same structure look like about 12 to 24 hours after the cyclone hit. So basically only the roof that you can see there in the bottom image was still visible. And in this situation, you might imagine, well, if perhaps not all of the park flooded, and as this map shows, only the flooding was concentrated around certain portions of the park, and as we move further away from the lake and out of this floodplain habitat, that black polygon there represents the lake in the middle of the park, there may have been some refuges for some of these animals to move into. And that's kind of was our naive expectation. If they could move away, why wouldn't they just do that? But what we actually saw was some really interesting and somewhat counterintuitive responses from some of the animals. And I just want to zoom into the responses of one species in particular, and that is a small woodland dwelling antelope known as a bushbuck. And I'll also come back to them a little later in the talk. And what we saw in this population, for which we had a subset of individuals collared, there was this strange bifurcation in response where some of the individuals unfortunately died, but some of them survived. And it turns out that this division in mortality wasn't kind of random act of chance. It wasn't about where they were placed on the landscape relative to flooding. It actually had to do with the behavioral decisions that those animals made at the time that the flooding hit, and specifically at how they changed their patterns of habitat selection. So what we looked at was whether these antelope selected for areas of higher elevation across the landscape and to try and escape the areas that were most heavily flooded. And what we found is the individuals that died, which on the left-hand side here, actually showed no significant change between their patterns of selection before and after. So the purple and gold bars are very similar, not significantly different from one another. But the individuals that survived showed quite a difference in their patterns of selection. So following the flooding, individuals that survived increased their selection for higher elevations, and they also marginally increased their selection for termite mounds in the landscape, which in Gorongosa, these kinds of small islands of high elevation refuges dotted across the landscape. And this immediately prompted me to ask, why? Why on earth would these animals make different decisions? Where did that come from? And obviously when the consequences were so severe with some individuals dying, why didn't they all make the right decision and move out of the way of the flooding? And these kinds of differences among individuals in behavior in the same population is known more broadly in the field that I work in, behavioral ecology, as individual behavioral variation. And this has actually been studied for quite a long time, over the last several decades, across many different taxa throughout the animal kingdom. And what this research has shown is that, similar to the case of the bushbuck behavior, it can actually have really important implications for individuals, for how they interact within the ecological community, and how populations can respond to different environmental conditions, particularly when they're really stressful, like what happened when the cyclone passed over Gorongosa. And to give you an example of a totally different context in which this can be important, studies have shown that for sea otters, when they live in resource-stressed environments, so resource-poor environments, individuals will actually start to differentiate from one another in terms of the diets that they eat. So different individuals will start to specialize on different diet foods in order to take better advantage of the limited resources available. And obviously, this differentiation is going to impact the individual nutrition and survival of those sea otters, but it's also going to have cascading influences on that prey community and the overall dynamics of those ecosystems where sea otters are a keystone species. And so this concept of individual behavioral variation preoccupies most of my research, and I tackle it through the specific lens of animal movement. And that's because movement is one of the most ubiquitous behaviors in ecology. It's really central to many other processes like foraging, hunting to acquire food, finding mates, or avoiding becoming someone else's food. And where, when, why, and how an individual animal decides to move will impact its own individual survival, its patterns of space use, but it will also scale up to impact population distributions and dynamics, community interactions, and ultimately even some key functions of ecosystems. And because of the central nature of movement to all of these other ecological processes, we might expect that any individual differences we observe in animal movement decisions could have particularly important consequences for populations and also for how we think about managing and conserving these populations. So why might individual variability in animal movements arise? Well, let's break it down and start with a very simple example. So say we observe an animal moving. In my case, this might be a pronghorn walking along the side of a highway as I drive home. In your case, it may be it's a deer or a black bear crossing the road as you're driving through the landscape. And the potential causes of that particular behavior occurring in that time and in that place can be really wide-ranging for any given individual. So it can be things like the animal's environmental context, the behavior of conspecifics or competing species, where predators are and what they're doing on the landscape, the animal's energetic or reproductive state, its age and experience, and also its spatial memory and knowledge and familiarity with its current environment. So in addition to all these different causal factors, the consequences of what decision that animal decides to make can be really wide-ranging. As I mentioned, they'll affect individual patterns of space use and individual survival, as well as reproductive success. But individual decisions can also scale up to impact the dynamics of populations, species distributions, and ecological community interactions. And so my research aims to tackle the full complexity of that picture I just showed you, and to address the central question on the ecological causes and consequences of individual differences in the movement behavior decisions of wildlife. And we then try and take this mechanistic understanding of these decisions and apply this to more informed wildlife management policies. And right now, in this particular moment, is a really exciting time, as I'm sure many of you are aware, to study these kinds of questions in the context of animal movement. And that's thanks to a whole range of different technological advances that mean we can get more fine-scale, high-resolution data on animal movement decisions than have ever previously been possible. So this is things like high-resolution GPS and remote sensing data, but also accelerometer data, better climate, and other environmental data. And what this fine-scale data allows us to do is get a really detailed picture of how animals are interacting with their environment and how they differ from one another. So this video is just showing three movement tracks of three antelope in Gorongosa. That's that same lake that I pointed out previously. As you can see, we get a sense of how these animals are using their environment. And when we break this down into individual movement tracks, we actually get a really detailed picture of how they're differing from one another. So these are the movement tracks of two different bushbuck, both living in Gorongosa in the same environment, in the same population. But you can see that with this fine scale data, we're actually able to tell how they're using the landscape and how they're differing from one another in their decisions. And we can break their movements down into different behavioral states and get a really good picture of the different ways in which they're exploiting their environments, and then try to understand why they're doing this and what the consequences of it are as well. So today I'm going to talk about how I have addressed just some elements of this picture in some of my research, and I'm going to move across two different study systems, African savannas, and then moving back to the Mountain West here in North America. And across these systems, I've focused on how predators, conspecifics, energetic state, and environmental context impact the movement decisions of wildlife. And I've examined how these individual decisions scale up to spatial distributions, as well as the ecological interactions within a community. And today I'll focus on just three of these projects. And throughout this research, as you'll see as I move through the talk, I've also worked closely with managers both in Africa and in my work in Wyoming to try and design it effectively around management priorities and make sure that our findings can be translated directly into ongoing management decisions. So much of my research in African savannas has focused on understanding the underlying factors that result in differences among individuals within the same population in their patterns of habitat use across the landscape, and specifically trying to establish a connection between the mechanisms underpinning these individual decisions and the consequences for the ecological community in these savanna ecosystems. And I've conducted these studies specifically on the large mammal community in Gorongosa National Park in central Mozambique. So Gorongosa is located at the southern end of the Great Rift Valley, and it's made up of a variety of really interesting and diverse habitat types. So at the center of the park is Lake Urema, which you've seen before and is now shown here in white. This is then surrounded by a treeless floodplain grassland, which in the dry season is a completely open grassland plain, and in the wet season, it's typically completely flooded. And you saw the extreme example of that in the images from the cyclone. This then transitions into a more typical savanna woodland habitat, which is shown in the darker green color here. And that black box there just delineates a core area of the park where we've focused on understanding shifting patterns of habitat use within two different antelope species across the boundary between that savanna woodland in the bottom and onto this more open floodplain grassland surrounding the lake. And just to give you a sense of how different these two habitat types are from one another, That top image there shows a bushbuck foraging out on the floodplain. So you can see it's very open, very few trees out there. And then the bottom image shows a bushbuck foraging in that savanna woodland. And you can see it's maybe much more similar to some of the shrub landscapes here. It's quite dense bush in a lot of places. So in addition to being a phenomenally beautiful place to work, Gorongosa is also very ecologically interesting. So the first aerial surveys of the park were conducted in the 1960s and 1970s. And at this time, it was home to a diverse community of large herbivores, and these were dominated by mega herbivores like elephants and hippopotamus. Now, unfortunately, during the Mozambican Civil War, almost all of these large mammal populations were nearly extirpated from the park. And these are the results of the first aerial surveys conducted in the park following the conclusion of the war. In much happier news, however, a very successful ecological restoration project was undertaken, and we now actually see biomass recovery to the point where the large herbivore community is approaching biomass levels close to what they were prior to the war. However, you'll notice that the community composition is now highly asymmetric. So instead of being dominated by mega herbivores, we now have a large herbivore community dominated by mesoherbivores, and these are more mid-sized antelopes, things like zebra, wildebeest, and waterbuck, which you may not be too familiar with, but I'll come back to Waterbuck in just a few minutes here. It's also important to note that the large carnivore populations were also almost extirpated from the park during the Civil War, but they did not recover in the same way as the large herbivores with this restoration process. So at the time of the research in Gorongosa that I'm going to talk about today, the park was still functionally a predator-free landscape. So there were lions, but they were existing at less than 50% of their densities prior to the war, and other species like leopard, African wild dog, and spotted hyena were present only transiently in the park. So in the context of this recovery of the large herbivore population, we observed two interesting and somewhat concurrent shifts in habitat use, in which one of the antelope species shifted or expanded their habitat use from the savanna woodland onto the floodplain, while another antelope species shifted their habitat use from the floodplain habitat into the savanna. And the first of these species was bushbuck. So coming back to that small size antelope that I mentioned at the beginning. So bushbuck are a very aptly named species. They are a woodland dwelling antelope. They feed on woody plants, and they prefer these kind of high cover habitats in order to avoid being detected and then eaten by predators. So they're really not very good escape artists. They need to remain hidden in order to avoid being eaten. And typically what this meant in Gorongosa is that their distribution was historically restricted to that savanna woodland habitat. So historic records from the park show that bushbuck were never detected out on the floodplain prior to the war and this recovery of the herbivore population. However, what we observed once this population began to recover following the war is that some of these bushbuck individuals, shown in orange here, and maybe here for locations, were actually now spending a lot of time in this open floodplain habitat, which is sort of north or above that dashed line there. So much so that some individuals were actually moving all the way out to the edge of the lake to forage. At the same time, the waterbuck population were shifting their distribution in the park. So historically, waterbuck were actually the opposite of bushbuck, and their distribution was restricted to the floodplain. And that's because they're also a very aptly named species. They prefer wetland or flooded habitats, so typically we're clustered around the edge of the lake there. What we observed in this population was a steady expansion into the savanna woodland habitat, which is shown in the brown here. It's also hard to miss that this expansion was accompanied by a huge explosion in the population size and density of waterbuck in Gorongosa. And I'll just leave that as a little teaser for now. I'll come back to the dynamics of that situation later. So these two concurrent observations were really interesting. Seemingly, both these species experienced a similar disturbance. Their populations crashed, they recovered, and they were doing all of this in the absence of their main predators, which were large carnivores. So we conducted two different studies to try and understand what the different mechanisms were underpinning these shifts in habitat use in these two populations, and then connected these mechanisms to the broader impacts on the ecological community in Gorongosa. So beginning with the bushbuck study, first, we investigated how they expanded their habitat use into the floodplain. And in this case, we actually relied on a large body of existing ecological theory to inform our hypotheses and our experimental approach. And specifically, we focused on the absence of large carnivores in Gorongosa at this time. So large carnivores are known to have a disproportionate impact on their ecosystem by the initiation of trophic cascades. And what this means is a large carnivore like a lion will consume a lot of its prey species like an impala, and this will then have an indirect impact on a lower trophic level via their prey species because it will alter the patterns of herbivory on, for example, the savanna plant community. And this is what's known as a density or numerically mediated trophic cascade. However, more recent research has also shown, in addition to these density or numerically mediated effects, large carnivores can also have an impact on the behavioral decisions of their prey species. And studies have shown that in order to minimize their risk of predation, some large herbivores will change their patterns of activity, perhaps being more active at night instead of during the day, or vice versa, or they'll use alternative habitats where the risk of predation is lower, or potentially they'll use some combination of these two mitigating behaviors. But what's still not quite clear, and well, at the time of the study was not quite clear in the large mammal literature, was whether or not these behavioral changes also lead to these cascading effects on lower trophic levels, like the plant community that these large herbivores feed on. And so it was this kind of gap in the literature that led us to our first hypothesis, and that was that the loss of large carnivores in Gorongosa was actually facilitating the shift in the habitat use of bushbuck and allowing some individuals to become fearless and move out into that open floodplain habitat. And to test this hypothesis, we used a series of predator simulation experiments in which we deployed both sham control cues and predator cues using sound and scent cues in combination. And then we paired this with GPS monitoring of bushbuck behavior. So we collected data from each bushbuck every 15 minutes, so we could monitor at a really fine scale how they were reacting to the simulated predator presence on the landscape. What we expected to happen was that when we simulated predator presence on the landscape, all the bushbuck would show some kind of avoidance behavior, want to move out of that area. But we expected to see a stronger response in the floodplain individuals, and that they would actually revert back to more typical patterns of bushbuck habitat use and increase their use of woody cover in order to try and mitigate what they perceive to be predators being reintroduced to the landscape. So now, beginning with some of our results, we first looked at that avoidance behavior. So in the sham control trials, what this plot shows is whether or not, on average, bushbuck avoided the area where we deployed cues. So in the case of the control trials, we saw no difference on the floodplain, and intriguingly, in the savanna, individuals got slightly closer to where we deployed the cues. And we do have a couple of hypotheses as to why that might have happened. So if you're interested in that odd behavior, then please ask me about that at the end. But in the predator trials, the response is much more consistent, and we saw that individuals avoided the area where we deployed predator cues, suggesting they tried to get away from that spot in order to mitigate the risk of predation as they perceived it. We then looked at whether or not individuals changed their use of tree cover and were using more typical patterns of bushbuck behavior. And in this case, in the sham control trials, we found no change across either habitat type. But in the predator trials, we found no change in the savanna individuals that were already in woodland habitat, so didn't really have much capacity to increase their use of woody cover. And the floodplain individuals increased their use of woody cover and did actually revert back to those more expected patterns of bushbuck behavior. So what these experiments suggest is that when predator presence was reintroduced or artificially reintroduced to the landscape, bushbuck did return to more typical patterns of behavior, which indicates that it really was most likely the absence of large carnivores in the system that was facilitating that shift in habitat use in the first place. So our second hypothesis was more focused on whether or not this shift in habitat use was also resulting in further differentiation in this population. So the floodplain and savanna habitats are very different from one another. Was the bushbuck living in each area actually also different in terms of their diet composition, their diet quality, and also their body condition, given that they may be feeding on different diets? And to test this hypothesis, we used diet composition and quality analysis, which relied on DNA metabarcoding of individual fecal samples taken from each of our collared bushbuck. And then we also took a range of different body condition measurements, and we did this at the time that bushbuck were immobilized to have the GPS collar attached to them. And in this case, we expected to find some differentiation between these two groups. What we found, first of all, is that they were indeed eating different diets. So this is an NMDS plot, which essentially shows each of our individual diet samples from the DNA metabarcode and the fecal samples, And the points that are clustered closer together are more similar in their diet composition. Those farther apart are more different in diet composition. And we were able to identify distinct floodplain and woodland savanna diet clusters. The quality analysis then showed that the floodplain diets were higher in digestible protein and digestible energy. And even more intriguingly, the body condition analysis showed that floodplain bushback tended to be larger in size than the individuals in the savanna. So all this together suggests there is a real resource advantage for those individuals that were becoming fearless and moving out into the floodplain habitat. So finally, we tested a third hypothesis that relates specifically to those cascading effects potentially of the absence of large carnivores on the lower trophic levels like the floodplain plant community. And we expected to find some measurable downstream impacts. We tested this using a herbivore exclusion experiment in a caged, uncaged paired design. And because we had that DNA metabarcoding analysis of what the bushbuck were eating, we could target this specifically to a floodplain plant that only the bushbuck were eating in any substantial amount. So essentially what we did was went out to the floodplain, found a whole lot of this plant, took a bunch of measurements on different aspects of productivity, then put cages around some and paired those with plants that were left open. The ones that were open were free to be eaten by bushbuck, and those in cages were protected from being eaten by bushbuck. And we went back a few weeks later and retook all of our measurements and compared what happened. So the plants that were kept in cages and that couldn't be eaten by bushbuck did much better. They actually had fewer of their stems eaten, they had more leaves, they had more flowers, and they tended to be taller in height as well. So all of this suggests that the bushbuck on the floodplain were having that measurable impact on this plant community. So to summarize results from this first study, we found that the absence of large carnivores was most likely facilitating this bushbuck shift in habitat use onto the floodplain in Gorongosa. We found there was a real advantage for those individuals that did move out into the floodplain and showed that fearless behavior in terms of diet quality, body condition. And then that third experiment was able to demonstrate those cascading effects and provide mechanistic evidence that behaviorally mediated trophic cascades can occur in large mammalian systems. Okay, so moving now from the bushbuck to the waterbuck. I apologize in advance if I mix those two names up because they're very similar. So in this case, as I said, these populations were in this similar environments. They were both experiencing an absence of their main predators. They were recovering. We actually had quite a different hypothesis about what we thought was going on in this population compared to the bushbuck. And it has to do with that huge explosion in population growth and density that I alluded to earlier. In particular, we had a feeling that what was going on in this population had something to do with intra-specific competition and density dependence on the floodplain. So I've already mentioned this dramatic change in species composition that we've observed in the recovering community in Gorongosa. But what I didn't mention was that among these mesoherbivore species, waterbuck are overwhelmingly the dominant species in this community. So these plots here just show the density trajectories of different large mammals or large herbivores in the Gorongosa community. The far left-hand plots are prior to the war, and the others are all after the war. And you can see that some populations have slowly recovered, others have kind of stagnated at low numbers, but the waterbuck down the bottom left-hand corner here has steadily increased. And actually, you probably can't read that axis there, but this population has exploded in growth such that waterbuck now exist at densities of over 30 individuals per kilometer squared, which is an unprecedented and pretty staggering number for this species. It's the highest it's ever been recorded anywhere across their range. And so what we expected or predicted that might be going on is that this population may actually, at these high densities, be starting to approach some kind of carrying capacity or limitation on the floodplain, and that some individuals were starting to move into the savanna in order to mitigate the intense density-dependent effects of intraspecific competition on the floodplain. So to test this hypothesis, we examined a series of specific predictions, which I'm going to just talk about only a few of these today in the interest of time. If you're interested in what else we looked at in this population, then please feel free to ask me about it later, because there was a lot more to the study than I have time to go into today. But the main question we were interested in the case of the waterbuck was trying to identify, first of all, was it density dependence that was leading this population to expand their habitat use? And if it was, how were waterbuck actually doing this? How were they changing their behavior and their ecology through their diet in order to be able to exploit the savanna woodland habitat, which is so different from their preferred floodplain habitat? So first we used some mapping and time series of density to verify that this expansion was actually taking place. And so as you can see, historically, the Waterbuck and Gorongosa were restricted to the floodplain, and then they've steadily expanded into the savanna habitat, especially in 2016 and 2018. And you can see in the density plot that the increase in density in the savanna started around 2012, and this was going on at the same time the density of individuals was still increasing in the floodplain as well. So secondly, we then tested whether Waterbuck were actually experiencing density-dependent effects on the floodplain. And what we found is that they were severely depleting resources in this habitat. We were able to do this using some much larger cages here, some larger herbivore exposures on the floodplain, where we paired a large herbivore exposure that kept Waterbuck out with an open control plot where Waterbuck could forage freely. And what this experiment showed is that those exposure plots had higher plant biomass, so higher productivity. In general, though, there was some variation in this pattern, mostly to do with fluctuating patterns of rainfall throughout the study period. But I think what's even more striking are these images on the right, which show the vegetation left inside the exposures and just how scarce and denuded the vegetation is on the rest of the floodplain, where Waterbuck were really eating everything right down to the roots. So this evidence supported our prediction that it was density dependent effects on the floodplain and intense intraspecific competition that was prompting individuals to shift into less preferred savanna habitat. Our next prediction was that we would again, similar to the bushbuck, see differences in diet composition and quality for the Waterbuck between these two different habitats because they are so different in nature from one another. And in this case, we also used DNA metabarcoding, but we had over 100 different samples from individual Waterbuck, and these were collected across 4 different years. And so these plots are the same style NMGS plots that I showed you for the bushbuck. So each point is a diet sample taken from a fecal sample from a Waterbuck, and those more similar to one another are closer together. And here we were able to identify distinct floodplain and savanna diet clusters, and this pattern was consistent across the 4 different years of data collection. We then combined this DNA metabarcoding with some measures of plant quality and food plant functional traits to see whether there was a difference in the quality of the diets between these 2 different habitats for the Waterbuck. And in this case, we expected the floodplain diets to again be higher quality, but specifically in the case of the Waterbuck, because this was their preferred habitat, they typically didn't spend time in the savanna prior to the war. So we took a lot of different measurements, but 2 of the most striking is that the floodplain plants were higher in digestibility and digestible protein. And again, this pattern was consistent through time. So the individuals that were moving into the savanna were eating different and lower quality diets. This then prompted our next prediction to try and understand, since some Waterbuck are moving into the savanna, how are they dealing with this lower quality resource environment? Maybe they're changing their behavior or their foraging patterns in some way in order to compensate for the fact that they're eating a lot tougher and lower nutrition diets. And excitingly, we were able to test this prediction at a really fine scale, thanks to a fun collaboration with the Criticam project at National Geographic. And what this meant was that we collected really fine scale, every 5-minute GPS location data from Waterbuck across the 2 different habitats. This was paired with accelerometer data. And then the Waterbuck also wore a camera around their necks. And so I'll just give you an example of what a Waterbuck's eye view of the floodplain is. If you get motion sick, maybe don't watch because it's very dizzying. So what we were able to do was take this fine scale GPS data and this accelerometer data, feed it into a behavioral classification algorithm, and you can see the output of that algorithm running down the bottom of the video here. Sorry, it's a little bit guilty. But what the really nice thing about this study was because we had access to these videos, we could see actually what the Waterbuck was doing at any given moment. We could have really high confidence in our classification algorithm because we could look at it and say, okay, the algorithm says the Waterbuck is eating. Let's go to the video. Is it actually eating? And we could iterate that process to make sure that it was really an accurate activity classification. And the accelerometer data allowed us to see when it was putting its head up versus down and get at that eating versus just walking around. Okay, that's probably enough for that fuzzy chin. So we took these behavioral classifications and created activity budgets for each of the collared Waterbuck. And we then looked at whether Waterbuck across the floodplain and savanna habitats for allocating time to different activities in order to kind of cope with the different resource environments that they were living in. And what we found is that individuals that spent most of their time in the savanna, so less time in that open grassland habitat, were actually spending less time eating compared to individuals on the floodplain. And this was a little counterintuitive at first, but I would think it has to do with the ruminant digestive physiology of these animals. So when consuming tougher diets, these typically require more time to process through the gut in order to absorb the necessary nutrients. So the waterbuck in the savanna were possibly spending more time ruminating, less time actively eating, and consuming more nutritious forage compared to the individuals on the floodplain. Now, similar to the bushbuck, we also looked for differences in body condition. And in this case, we had kind of an interesting result where this body condition appeared to be similar across the two different habitat types. So possibly the lower quality resource environment in the savanna was kind of offset by the intense competition that was going on in the floodplain. And then using, again, the GPS and video data, we were able to estimate daily energy budgets for the waterbuck across two different habitat types. And in this case, we found something quite interesting in that waterbuck on the floodplain, despite competition out there, was still maintaining a higher daily energy balance on average compared to the waterbuck that had moved into the savanna. So what this indicates is that although waterbuck were able to alter their behavior to cope with the resource environment in the savanna, it may not actually be a viable long-term refuge for them because they weren't maintaining as positive energy balance as individuals on the floodplain. So to summarize results from this study, we showed that severe resource depletion was prompting waterbuck to move from the floodplain into the savanna. And in order to cope with the lower quality environment on the savanna, waterbuck were flexibly changing their behavior and their diet selection. Okay, so now bringing these two studies back together, how do these two seemingly quite similar and concurrent patterns of habitat use compare to one another? So by focusing on the mechanisms that were underpinning these shifts in habitat use, we found that bushbuck on the one hand were expanding their habitat use in the absence of their main predators, becoming fearless, whereas waterbuck were being prompted by intense density dependent effects to expand their habitat use into historically less preferred environments. And what this suggests is that overall, these patterns of habitat use in the bushbuck were kind of being prompted out of more of an ecological opportunity presented by the absence of their predators, whereas waterbuck were perhaps acting out of more of an ecological necessity in order to mitigate intense interest-specific competition. And the effects on the broader ecological community also differed, interestingly, between the two. So in the case of the bushbuck, we found those downstream cascading effects on the floodplain plant community. And in the case of waterbuck, we found that their flexibility in diet selection and behavior enabled them to tolerate a lower quality resource environment and potentially continue to grow and expand in Gorongosa. And in terms of the community dynamics, what this meant is that waterbuck might perhaps be actually suppressing the recovery of other close competing species like wildebeest and zebra that are similar in size and have similar diets, grazing diets to waterbuck. But our energy budget analysis showed that although waterbuck are able to occupy the savanna, it is somewhat of a marginal habitat for them energetically. So perhaps as populations like zebra and wildebeest continue to recover in Gorongosa, the waterbuck in the savanna may not fare as well in competing with these other large grazers, and it may not be a viable long-term refuge for this population. So there may be some hope that the community composition may return to something more closely resembling its pre-war state. And finally, because we focused on the specific mechanisms underpinning these changes in habitat use behavior, we can make some specific conclusions about what these behavioral changes meant for management decisions being made in the park. So the basic goal of the Gorongosa Restoration Project is to return the park, and specifically the large mammal community, to something resembling its pre-war state. And in the case of the bushbuck study, what we found is that in order to restore species interactions and patterns of habitat use and behavior that existed prior to the war for the large herbivore community, it was likely going to be necessary to reintroduce large carnivores into this ecosystem. And really excitingly, Gorongosa has undertaken a very extensive large carnivore reintroduction program where they now have very healthy populations of African wild dog, leopard, and they've just reintroduced one clan of spotted hyena as well. So it's a really interesting environment ecologically right now. In the case of the waterbuck, at the time, managers in the park were really kind of concerned with what was going on with the waterbuck population, and that there was just so many of them, and maybe they were suppressing the recovery of other close competing species. They'd so far taken a very hands-off approach, and we're starting to get to the point where they're wondering if we needed to take a more hands-on approach, potentially cull or remove some of this population. And what this study showed is that actually Waterbuck was starting to exhibit some signs of incipient population regulation. So they were starting to experience density dependence on the floodplain, and this kind of reassured managers that they could probably continue with their hands-off approach, which was their preference, because Waterbuck may actually be starting to regulate themselves. And since the conclusion of this study, we've seen other signs that this is indeed what's going on with this Waterbuck population. Okay, so now I'm going to make a big gear change and shift back across to North America to talk about, again, individual variation in the movements of large mammals. But in this case, instead of habitat use, I want to focus on what I think is the other aspect of large mammal movements that are so important for their population dynamics and for their management. And that is their large-scale movement decisions, like whether or not they're going to migrate, remain resident, or perhaps be nomadic in their movement behaviors across the year and across different seasons. And specifically in this study, we wanted to look at how variability in the environmental features of the landscape in which animals live can impact which of these movement strategies they decide to use. And we focused in particular on variability in the landscape of different habitats across the state of Wyoming, and on the movement strategies of pronghorn across this state. So there is a long-standing body of theory on animal movement strategies, and it suggests, or it makes very specific predictions about the types of environments where each of these strategies is expected to evolve. And this has to do with the degree of spatial heterogeneity and temporal predictability of those environments. And a lot of empirical research has tended to support this theory by looking kind of at environmental variations across different ecosystems. And what it's found generally is that migratory populations tend to occupy environments that are highly heterogeneous in space, but very predictable in time. So for example, high plains and mountainous environments that experience large seasonal fluctuations in resource availability. And similarly, nomadic individuals have tended to be found in environments that are highly unpredictable in space and time, and where resource availability tends to be more scarce. These are often dryland ecosystems, like the steppe ecosystems in Mongolia. But this advent of movement and high resolution tracking data that I mentioned at the start of my presentation has really revealed some interesting nuances to how these movement strategies play out across different populations and ecosystems. And what we found is that actually, despite this connection between the environment and movement strategies, that individuals within the same population, so seemingly living in the same environment, or at least in very close proximity, can actually exhibit all three of these different movement strategies. And so this kind of runs in contrast to our theoretical expectations. And just to give you an example of what this looks like, these are three movement tracks from three individual pronghorn that all live in the same population in Wyoming. So you can see at the bottom of the map there, they share a winter range, but then throughout the year, they each exhibit a different movement strategy. One's migrating, one's pretty nomadic, and the other remains resident in the winter range throughout the year. And so our objectives with this study was to try to reconcile these kinds of observations, which are becoming increasingly common in these large ungulate populations, with that existing body of movement theory. And so first we wanted to just quantify how diverse pronghorn movement strategies across Wyoming actually are. So if that one example is just one good example that I managed to pull out, or if they really are very variable. And then secondly, we wanted to test whether these same variables of heterogeneity in space and predictability of the environment and time could still explain some of the variability in these individual movement strategies, even when we zoom in to very fine spatial scales among individuals living in the same population in very close proximity to one another. And we focused on 8 different populations of pronghorn from across Wyoming, and these are just our different study populations. And you can see they're distributed throughout a lot of the state and in quite different environments. So the first challenge with this study was to try and define individual movement strategies. And this was tricky because we were trying to place individuals not just as migratory or resident or nomadic, but between all three strategies using the same metrics. In this case, we were not able to come up with just one, and instead settled on using 2 metrics that, when we consider them in tandem with one another, do allow individuals to be placed in one of these 3 movement categories. And so this is the same map showing our study population, and each of these circles represents an individual, the circle is centered at the centroid of each individual's home range. And I'll just go through and explain a little bit more about what each of these metrics are actually showing. So the size of the circle corresponds to year-round overlap in space use for that individual. So how much an individual is staying in the same place or moving around throughout the year. And this corresponds to a relative nomadic to resident tendency, where nomads have very low overlap year-round and residents have high overlap in their space use patterns year-round. The color of the circle then corresponds to a resident to migratory tendency. And this is measured using the overlap between summer and winter space use. So how similar their summer and winter ranges are, residents would be more similar or overlapping with one another. Migratory individuals are expected to have no or very little overlap between those two ranges. And so when we look at these together, just to give you an example, the large dark purple circles are individuals that have high overlap year-round and between summer and winter. So these are residents. And then migratory individuals tend to be more intermediate size circles, so intermediate levels of overlap year-round, but darker orange in color because their summer-winter overlap in particular is very low. So the next component of this study, which was also quite a challenge, was figuring out how to quantify the variability in the environment. So in this case, we use a whole range of different environmental variables, including some of these ones listed here. I'm not going to go too much into the details of this, but if you're interested in our methods and more information, then definitely ask me about it at the end. But we use these variables to come up with first a measure of temporal predictability. And this is essentially a variation in the environment from year to year. For example, how different is the date of the peak green up in spring from one year to the next? And then secondly, we looked at spatial heterogeneity, which is more within year variation in the environment. So for example, how much are the habitat or environmental variables changing from one, if I'm here in 1 kilometer grid cell, I move to the next 1 kilometer grid cell, how similar or different are they to one another? And these images here just give you a sense of how these two variables combine to give rise to different types of landscapes. So the mountains and plains that I mentioned, more dryland ecosystems. And in the case of pronghorn, they occupy both these types of environments. So you can start to see how they might start to become so variable in their movement strategies. So we used beta regression models to test the relationship between these environmental predictors and individual movement strategies. And we used both spatial heterogeneity and temporal predictability in the same model with each of our two movement strategy continuums, so migratory to resident, resident to nomad. And what we found is that they were significant predictors of individual movement strategy. And when both were included in the same model, we were able to explain about 30 to 40% of the variability in individual movement strategies using just these environmental variables on their own and not considering anything else about these individuals. And when we consider all these variables together on the same plot, we start to see some kind of quite interesting patterns emerging. So we found that migratory individuals occupied these highly spatially heterogeneous but temporally predictable environments down the bottom to the right-hand side of the spot, whereas residents tend to occupy spatially homogeneous environments across quite a wide range of environmental predictability and time. And then nomads occupied that intermediate space between these two. And what was really exciting for me about this study was that we could then take the information we gained from roughly these 400 pronghorn and start to predict the movement strategies that we might expect to see across an even wider range of environmental conditions in areas of the state where we don't actually have GPS data on individuals. So we can try to extrapolate beyond the areas where we actually have collaring data. And this heat map just gives you an example of that, but we can also apply this directly to the landscapes in Wyoming and start to predict in different regions what movement strategies of pronghorn are we most likely to see. And this is really useful for managers. We're working on some of these predictive maps with Wyoming Game and Fish at the moment because the prescriptions that managers need to use for migratory, nomadic versus resident individuals are actually very different from one another, but they also can't collar every single pronghorn in Wyoming. So being able to make these kinds of inferences helps them make more informed decisions about how to manage in different regions. So coming back to that original theory, how did we actually match up with our findings? Well, we found that migratory individuals tend to occupy actually the exact type of environment that you would expect. So it's clearly strong support for that theory. But we found that resident individuals occupy a much wider range of temporal predictability than we would have expected. And this is actually some of the landscapes where we would expect, according to theory, to find nomadic individuals. So while this work could resolve some of the complexity and the environment can clearly explain some of the variability in individual movement strategies, it leaves kind of much more to be explored, and actually, for me, raised a lot more questions than it answered, which maybe is frustrating, but for me, it's actually one of my favorite parts about what I do in studying these kind of fine scale variability in animal behavior, is that often when we look at this, it reveals more that we don't know about these animals. And it's these kinds of questions that continue to drive my research forward in order to really answer this question, the central question, in a comprehensive way, and in a way that encompasses all of these different causes and consequences, as well as the full scope of the different behavioral decisions that animals are making. And so at the moment, I'm just developing my research program at Montana State, and we're currently developing some new research directions in Montana and continuing our work in Southern Africa to try and address all of these components in an integrative way. And ultimately, the aim of our research is to use this mechanistic perspective on individual behavior to enable its application as more of a window and a tool for the assessment of population status, ecosystem health, and ultimately to generate creative management solutions. And with that, I just want to sincerely thank all the collaborators that contributed to this work. It was an unbelievably interdisciplinary and team effort to get all of this work done, including actually two of the folks here today, which is exciting, and also all of our funding sources as well, and all of you for attending, and I'd be happy to take any questions. Thanks.
So you can about the mechanism for this movement, like choices or categories of the compound. So was it consistent across years? So like individual inherent trait of the food or dominance dependent?
Yeah, that's a great question. That was my first question too. So we're actually doing kind of part two to this study. So in this case, we just looked at 1 year of data from each individual, took whatever they were doing in that year, and ran this initial analysis. And now what we're doing is going back to those individuals that we have multiple years of data for, which is actually, unfortunately, about 10% of all of them. Pronghorn are pretty sensitive species, so it's hard to follow them for too long. But we're going back and looking at what individuals do between years and whether they're switching. We think switching, at least in pronghorn, likely has a lot to do with environmental conditions in the previous year. So they're very sensitive to winter and especially snow conditions and resource availability in particular. So we think in their case, it probably still has a lot to do with the environment, but in a different way. We know a lot less about how or whether they learn movement strategies from one another than we do compared to other ungulate species. I think we have a much better idea, for example, in mule deer, that there's probably some kind of matrilineal association going on there, but with pronghorn, we're just starting to scratch the surface of how they figured that out in addition to the environment, but there's likely a large cultural component as well, I would guess.
Yeah, so also, I was curious if it, in the matter of statistical, migrators.
Yeah, I think the scale component that is really important. So we were measuring the environment at about a 1 kilometer grid cell, basically because that's the GIS the remote sensing data we were working with. So at larger spatial scales, I'd actually expect those relationships to be stronger because you tend to see more clustering into environments that are clearly migratory landscapes like the mountains and plains, and those that are clearly more predictable, perhaps in space, and so more resident. And that's kind of what other studies have shown, looking across different countries or different ecosystems where those populations aren't interacting with one another. I think at an even finer scale, we would probably see a lot more nuance because we'd actually see how the pronghorn, like I was just saying, are interacting with snow and making those finer scale decisions. And one plot I didn't show, but what we've looked at is these movement metrics were kind of continuous, but we also tried to kind of classify individuals into discrete strategies based on different cutoffs. And what we found is that the vast majority lay somewhere in between one of those three strategies. And probably a lot of what that has to do with is that they're just being really kind of flexible and responsive to finer scale variation than we were able to quantify. But we're hoping to get much more fine scale snow data as we kind of advance our ability to get that from remote sensing, which is starting to happen. So I hope that answers your question. Sorry, I'm not sure.
Is there a sweet spot in terms of how frequently you're getting GPS fixes on these animals tracking? And you said that you've got multiple years of data for lots of things. So how are you balancing that?
Yeah, so I think in this case, I kind of showed a whole range of different fixes. It really depends on what question you're trying to ask and how much data you need to collect. So for the bushbuck, we really wanted to know at a fine level of detail how they were responding. So we took those the fix rate way down to 15 minutes, and that meant those calls were only out for about 6 weeks before the battery died. And same with the waterbuck, 5 minutes, they lasted like 2 weeks. With the pronghorn, most of these data came from more longer term studies that state managers and other agencies are running to try and basically monitor survival and recruitment and patterns of habitat selection in these populations. And so they were, I think most of these are on average 4 hours because they're trying to keep them out for at least a year. They do try to redeploy them as well and keep them out for longer, but yeah, pronghorn, if it's a bad winter, they have really low survival rates. And so that's why we don't have much. The intention is to have more longitudinal data, and it's just really, they're a tricky species to get that kind of data.
So the pronghorn are parents. So how does that interact with the movement strategy that they?
Sorry, I think I forgot to mention, these are all females, first of all, so that definitely plays a part. So we don't really know what the males are doing, although I suspect they're probably following what the females are doing. So most of these distinction in strategies generally has to do with what they're doing in the breeding season, or at least that's where the migratory behavior comes in, because they'll migrate in the spring towards their summer range. It's those that migrate. I don't know to what extent that has too much to do with exactly what the breeding system is, mostly because they're all females and so they're likely moving together. I think it probably created some of the kind of noise and variation that we see in strategies because some of the collared individuals are likely moving together and in the same social group. And so that's kind of gets back to your first question about the cultural component. I think probably that's why we're kind of, the environment's able to get at some 30 to 40% of the variation, but this is still this other big unexplained part. And I think that in large part has to do with the fact that they live in these groups, and if one individual in the group is going that way, then that's probably what they're going to do as well, regardless of the environment and what they're experiencing. There's actually another student in Wyoming who's trying to look at some of those questions about how they make decisions and how their social environment affects that. So I'm really interested to see what he's able to find, because we don't really have a good sense. So I can't answer your question very well, sorry, but hopefully in a couple of years we will know much more about what pronghorn are doing socially.
Well, I think there are some drinks and snacks that we can hang out, and let's thank Justine one more time.